Good morning, good afternoon, and good evening. It is with great pleasure that I welcome you all today for this webinar on optoacoustic imaging in neuromuscular disease. I'm still Dr. Tim Devling, Director of Sales and Applications at Ithera Medical, and I'll be doing the moderating today. The webinar will last for about 40 minutes, and we really encourage questions. If you maximize the GoTo tool, you can see a question box where you can type them in, and we will answer them at the end. The webinar will also be recorded, and we'll send you a link in the next few days. So it is with great pleasure that I introduce our speaker today, Dr. Ferdinand Knieling. Ferdinand is a true clinician scientist based in the Department of Pediatrics at the University Clinic Erlangen. The impact of his work has been well recognized, with the current focus on innovative diagnostic methods for neuromuscular disease, receiving the Adalbert Zerny Award in 2019 from the German Society for Pediatrics and Youth Medicine. So with that introduction, I hand you over to Ferdinand for the talk.
So also hello from my side, and thank you all for joining this webinar about optoacoustic imaging in neuromuscular disease. So before starting this webinar, I'd like to introduce you to the topics of our talk today, and it will go around the technical background of optoacoustic imaging and also the medical impact of imaging in this field, and then about our study findings, what we found out about optoacoustics in neuromuscular disease. And then I'd also like to show you some novel data and give you some outlook where this could go and where we see applications for such a technology maybe in the future. So with this, I'd like to start, and I'd like to show a more or less historical introduction of optoacoustic imaging, which started like way behind maybe in 1880, where Graham Alexander Bell described the photoacoustic effect, which is more or less the conversion of light into sound waves, and he used it to transmit information for telecommunication. And this was developed very much further by different people now evolving into an imaging modality. And here is maybe some of the first images derived by Liang Wang from in vivo imaging in a mouse brain. So how was this developed and how is this effect described? And this was already way behind. It was 1880 by Alexander Graham Bell. And as I told you, he used this phenomenon of transverting sound transverting light into sound waves for transmitting information of a certain distance. So what he did, he used the sunlight painted here and led it over mirrors and flexible mirrors, which are modulated by a mouthpiece to deliver the speech over into a parabolic mirror and in a selenium cell. And it was then reconverted into sound waves. And by this, he could transmit sound waves for telecommunication, but it did not end up in the end, but it was called the photophone. And if we take it like very shortly, it's light in and sound out. And this is the principle which we still use for this imaging methodology. And nowadays it's not polychromatic light, it's laser light which we are using. So we are using focused laser pulses, and if you emit these laser pulses into tissue, you kind of get absorbance by different molecules in the tissues, which then generate acoustic pressure waves. And these pressure waves travel very far in comparison to light, and you can then detect them maybe with a tomographic array and then reconstruct it. And you can also, if you use different wavelengths, do a spectral analysis and derive different components in the tissue by this. And this does not only work on the surface, this potentially also works with high spatial resolution and depths of centimeters. And I would say this is really clinical relevant depth compared to other optical imaging methodologies. So optoacoustic imaging is not like a homogeneous technical background. It uses different setups, so it can work with really high resolution for microscopy. But what we are talking about is here painted in C. It's non-focused waves and non-focused ultrasound, and this gives you higher penetration and also maybe not so good resolution, but still very sufficient to image different tissues. And we were not the first to describe this. And it's not a table which you should read from beginning to end, but it's just to illustrate that there are many clinical studies or many pilot studies looking at skin cancers, the skin vasculature, lymph nodes, inflammatory bowel disease, whatever. But there are many people researching in this imaging technologies. And one example is one study conducted by the group of Maximilian Waldner, and we looked at inflammatory bowel disease to detect the hemoglobin content, where hemoglobin is a very strong absorber for optical imaging. And as you can see here in this live video, you can actually detect and quantify the content of the hemoglobin in the intestinal wall, which can be a measure of inflammatory activity. And this was like the very start for us to look into this imaging technology. And we are not only talking about hemoglobins, which are shown here with the unique absorption curve, and you can also image maybe melanins from the skin or tumors, but you can also, as I've shown here, different types of oxygen saturation or saturated hemoglobin, and maybe there are much more absorbers in tissues which can be used then for imaging, which can be namely water, collagens, or lipids, and they also have unique absorption properties, and they could potentially then be separated in an imaging modality which covers the near infrared and also the extended near infrared range of light. And this is totally durable by MSOT system at the moment. So if we then look at the medical impact of the research we are doing, and also for neuromuscular diseases, we have to ask, do we really need new imaging technologies? Don't we have a lot, like many imaging technologies which are suitable also for children? And I think we have to share some thoughts about it. And it's really illustrated by some pictures drawn here by Giotto and also by Raphael, some years maybe between them. And you can look at children by the way of small adults, but we really much know that children are different. They are not small adults. This Jesus is like really a kid, a baby, and it has unique properties. So they have different diseases, they have different cause of diseases, they live longer, and they also have lower masses, which give them higher radiation doses if you want to use ionizing radiation. And we know their organs are much more sensitive to radiation. So in the terms or in the view of a pediatrician, it's very important to develop technologies which are suitable for these kind of patients. And here just drawn a huge setup of technologies which could be used to image maybe muscles and what we want to do. And if we look at safety and usability, and drawn here in red on the bottom, you see CT, MRI, PET, SPECT. They have maybe, I would say, a huge problem concerning ionizing radiation, also portability or scanning times. They all have huge efforts at this site. And if you compare to optoacoustic imaging, I think there are huge advantages on the side of optoacoustic imaging. And then if you look on the side of information which are provided, or which you can derive from such an imaging technology, you can see that by optoacoustic imaging, you get morphological, functional, maybe also molecular scale and dynamic information, which you can't get, for example, from a PET or a SPECT image. So I think this technology really has unique properties which we can use, and I show this later, for such neuromuscular diseases. And if we then look at neuromuscular diseases, and I think we have a broad audience, maybe some of you haven't heard of these diseases, it comprises very heterogeneous groups of diseases, which can either be directly related to a muscle injury or to a problem in the nerve innervating the muscle. So this is a really big group of heterogeneous diseases. And today I like to show you two examples of these groups. One Duchenne muscular dystrophy, which relates to a muscular dystrophy, so really a disease which happens directly to the muscle, and a motor neuron disease, which has secondary effect on a muscle. And I try to show you some results in these two fields. So when it comes to Duchenne muscular dystrophy, this is a disease where you have an absence of a specific protein which links the cytoskeleton to the muscle cell membrane. And if this is missing, you have a breakdown of the cell and you have degenerating muscle cells, and you have a loss of physical abilities for these children. And these children are only male because it's an X chromosomal disease and it has actually high incidence. And the absence of this protein leads then to muscular cell necrosis. And in the end, it's progressive. And all of these patients get problems with their heart, or with their lungs, and ultimately they die from this disease. So here are just some MRI images from some muscles there, and you can see over time, this is a younger patient, this is an older patient. There's like really severely affected, and you can also see it on MRI images. So usually we don't regularly do, at some point we do, but regularly we do some physical testing on these patients, and these tests are also used in studies. And some of these tests are very easy. This is the 6-minute walking test, and you basically let these patients just walk for 6 minutes and count the distance or the covered distance by the patient. And this distance is kind of predictive for the disease, and it's also used as endpoint in pharmaceutical studies, which is a problem because I would say each of these tests, whatever they are testing, and if they are physiotherapeutic tests, they require a patient which is cooperative, and maybe this is not really doable in very young patients. So here's a video illustrating the natural cause of the disease, and what we like to show you is a patient and a like age-matched control on the left side, and they got the same thing to do. They should climb the stairs here, and you can see it's no problem for a healthy boy, but you can see the boy with the Duchenne muscular dystrophy on the right side. He really requires much more time for this, and this is just an example for one test we are doing. But of course, this test is not doable anymore in higher ages, as you can see on the right side, because they become wheelchair bound at a specific age, and then you have to kind of adapt your tests to this physical state. So there's the second disease I'd like to show you, the very similar test. It's a girl suffering from spinal muscular atrophy, and she was also asked to climb these stairs, and she's accompanied by our physiotherapist here, because you can see there's a high risk that she maybe falls or does not get one stair. And this disease is, like from a pathophysiology view, a little bit different, because there's a loss of motor neurons here, and this secondary leads to a loss of muscular mass and an atrophy, and then it also can lead to a loss of walking ability, for example. But however, this disease is also to some kind very frequent. It's 1 to 10,000 incidents, and we have a carrier frequencies in some groups of 1 to 50. But the spectrum of the disease can be very, very broad. So we have a prenatal onset, but we can also have a very late onset where the symptoms are very, very hard to discover and the diagnosis is not that clear. So if we move on, and I like to explain you a little bit about the pathophysiology in SMA. In SMA, there's a loss of a gene function or a gene. There's maybe a mutation there in the SMN1 gene, and the loss of this protein ultimately leads to degeneration of motor neurons. But however, we don't know why. There are some backup copies of this gene called SMN2 in a maybe little bit different fashion, and some of the protein which is produced there can still work. It's about 20, maybe a little bit lower percentage is still functioning and working as an SMN protein. And this is why there are different therapy strategies, maybe to replace one gene or to alter the expression of the SMN2 gene, which are currently developed and currently actually approved right now for these patients. But still, and this is the last clinical message here, which is very important, we don't know yet where the optimal therapeutic window is. And this is also true for Duchenne patients. Where is your option to treat them? We think it should be very, very early, but we don't have really good tools to monitor this. And as I showed you, for SMA patients, there are really different types and different causes of the disease. So you need to have some predictive tool to monitor this maybe over years. And as I told you, there are 2 medications for SMA approved right now. And this just last week or the week before, this therapy called Zolgensma was approved. It's very, very expensive, but it's a single dose gene replacement therapy. And it seems to be effective, and it's now approved in the US, in Japan, and also in other major markets like Europe. And we have another medication, it's Spinraza, it's an oligonucleotide, which alters expression of SMN2, and it's also approved for these patients. It's very, very expensive, I don't have to say this. So we need to have objective biomarkers to somehow predict if these interventions are really effective or not. So I'd like to show you now what we found out in patients with Duchenne, and we just got from the technical point where light in and sound out, I showed you this, got into some phantom studies. I don't show this today for the sake of time, but I'd like to show you how we went from disease models into patient and then into longitudinal studies with this pig model. So it's not only my work, so we have a lot of colleagues involved, especially Alex and Adrian, working on this topic. And this is just one picture we got there from our studies in Munich, where we worked with this animal model. And this animal model, it's a pig model. It's very, very interesting because it mimics much more closely the disease as maybe a mouse model today can do this. It's a deletion of a specific exon, which is comparable to the human disease, and we imaged these pigs just 1 to 3 days after delivery. And what we can see, you have the live image here on the upper screen, and what we can see there, we can actually quantify the signal of what we think is the collagen derived signal here in the muscles of these pigs, and it's much more increased already 1 to 3 days after birth compared to a normal wild type pig. And we had the chance then to also look at tissues, and if you look at a tissue level, you can see here on the H&E stainings that you get somehow a destruction of the muscle tissue, where some of the tissue is replaced here, stained in blue, by tissue which is, for example, can be collagens, but it's more or less connective tissue, and also here stained in red. And if you quantify this, the area which is stained as connective tissue here for the trichome or serous red staining, or also if you take this and measure the total collagen content by a hydroxyproline assay, you can see there that there's a really nice correlation of MSOT signals, which we derived, and the staining of the positive area or the collagen content. So that we see a pronounced fibrosis in these Duchenne pigs, and we see that it's really significantly higher than in wild types, and that we can quantify it, and it really correlates to what we did ex vivo. And as a next step, we took this to our patient, and we imaged 10 patients and 10 age-matched healthy controls. And you can see it already by these color-coded images. You can see there that there are huge differences at different muscle areas, which are the biceps, flexors at the arms, or the quadriceps or gastrocnemius muscles on the lower leg. And you can see there, you can quantify it. And if you quantify it, you also get these significant differences. If you calculate it over every point or for every patient, they are significant. And if you do a receiver operator curve analysis, which is more or less to count or to calculate specificity and sensitivity for a diagnostic test, you see that it's very, very good actually. And on top of this, you can also do it in a three-dimensional volumetric way. It's just a technical detail. If you use another detector, which is more like on a cup shape, you get volumetric information. You can also see it here also already in color-coded images in different projections here, and also in the volumetric view. And coded here in blue, it's the collagen, and in red, it's the hemoglobin or myoglobin signal. And you can really see here that there's a huge increase in the Duchenne patients. And if you quantify this, it's more or less similar results. You get really, really specific and sensitive results to detect patients with Duchenne muscular dystrophy. And on top of this, I would add, if you correlate now our findings on 2-day or 3-day images to a diagnostic test, then you can see a 6-minute walking test really has a nice correlation to our imaging findings, and also a 10 meters run, or as I showed you, these 8 stairs climb. It really correlates to what we found on imaging. And then at the very end, we were asked, can you reproduce your results also in a longitudinal fashion? Which means, would you be able to monitor the progression of the disease? And yes, we had the opportunity to have a follow-up on these pigs. And you can see here on the video, these pigs get much more active. It gets much more complicated after 4 weeks to have imaging from them. If you imagine, if you want to do MRI on them, it's not even possible to do without sedation. But for an MSOT, which takes only seconds, it's totally possible to do it also without sedation in the most cases. And you can see here over a course of 4 weeks, there's a growth of the muscle, but there's also huge growth of collagen signal in this muscle, which we then quantified. And you can see here there's an increase in these Duchenne pigs, but there's not an increase, there's stable signals in wild types, which means, or which we interpret that we could say, okay, at least we can see the progression of the disease, and maybe in the future it might become a tool to also monitor therapeutic intervention. So, and this is where I want to give you now a little bit of an outlook, and this outlook should also cover first that we reproduced our results in the collective of patients with SMA. I told you this disease is a little bit different. It affects the muscle, not directly, but secondary after the nerve, which should like control the muscle, is degenerating. Also, the muscle is secondary atrophic. But what you can see there also in the B mode, the muscle now looks different and also gets a different signal on optoacoustic imaging. While it looks very bright and homogeneous here in this 800 nanometer image, it somehow becomes destroyed here in the SMA patient. And if you look a bit lower here, already again colored in blue and yellow, it's a lipid and collagen signal. You can see here we see an increase in these signals, and we can also quantify this in this like preliminary analysis from these patients. But still we would say, okay, we have similar, a little bit different, but similar findings, and we think we can also differentiate these patients from healthy volunteers. And if we take this further again, and I painted here this together with the Duchenne patients, you can see if you do this also in a volumetric way, you get these results like very different from healthy ones, also in the SMA patients, where we have very, very bright signals for collagens, and also in some patients for lipids too, and you can quantify this, and there are also significant differences. And then, and I think this is a very, very important information for clinicians, we ask ourselves, are our results really reproducible? And we went back now last year to healthy people and to healthy volunteers, and we kind of tried to standardize a protocol where we imaged a certain protocol. It's painted here a little bit, and with 2 examiners within an interval of 14 days, and also included some exercises. And we wanted to know how much does a time interval, how much does an examiner, and how much does exercise affect actually our measurements. And what we can show you now is that we calculated the correlation, or something which is a clinical correlation. It's called the intra-class correlation, and it intra-class correlation coefficient, and it says how strongly does units in the same group resemble each other in the group. And this is a measure how good a diagnostic tool could work, and we calculated this over each single wavelength for the scan in the muscles. And as you can see here, you have different grading for like the precision of such a measurement, or how good it is. And you have like the degree of excellence, which is between 0.8 to 1. It's very similar to a simple Pearson correlation, for example. And you can see here it's pretty excellent actually for one rater. So if one rater measures repeatedly, he very much measures the same thing, we think. And also this similar thing is true for a parameter. So for deoxygenated, oxygenated hemoglobin, total hemoglobin, collagens, or lipids. So you really measure in a high reproducible way. And then it's a similar images. If you have two raters, for example, you get very similar results with a little bit more distribution, but still the results over the entire spectrum from 660 to over 1,200 nanometers, it's pretty much the same. It's stable, it's reproducible. We are very confident with our results that we are measuring similar things if we do it repeatedly. This is also true maybe with one thing which is different, and it's lipids. We don't know why yet, but maybe it's due to differences and in girls and boys, actually, how lipids are distributed there, but we don't know yet. We can just describe it, but as you can see, it's still moderate, but still we are confident that we are measuring things which are really true. So then we ask, and this is my last slide today, is this also reproducible about in a certain time interval? And we would also say, just from the images here, so this is pre-exercise and this is post-exercise, which what you maybe can see is that you get a little bit more signal post-exercise, but 14 days later the images still look similar, so that we can say at least we get values in the same range. At least for 14 days, our measurements are very, very stable, and we can say, okay, maybe this tool could work over a certain interval, but I think this is just a starting point, and this is the point where I like to wrap up my talk today. And I told you that optoacoustics, and I think it could have great impact in the clinics in the future, and I really think it's translatable to pediatric applications, especially for muscular imaging. But I think what I showed you at the end, we need more longitudinal studies. And with this, we really need technical works. We need our colleagues to address questions about fluence corrections for deep images, also to more confidently separate chromophores, and also on the quantification side. I think there's very much more to do. But with this, I'd like to end, and I also thank you for your attention. I'd like to thank many, many people which contributed in the last years, and I'm happy to take your questions. Thank you very much.
Thank you very much, Ferdinand. Now, we do have a fair number of questions have come in, so I'll start right now. First question is, you showed imaging of hemoglobin in inflammatory bowel disease. Would there be a reason why you couldn't image collagen in IBD? Would this represent fibrosis in IBD or other such diseases?
Okay, yeah, thank you very much for this question. This is actually more or less related to a time sequence we had. So we started off actually with inflammatory bowel disease, and the whole collagen story was a later project. So we're very interested to see disease progression also in inflammatory bowel disease. And it was just a matter of, let's say, time and focusing on one question. But yes, I know there are publications already out from different people showing spectroscopic data that there's like changes in collagens in inflammatory models, especially inflammatory bowel disease. So this is one topic which is already addressed also by many other groups, and there are already interesting results out there.
Okay, some technical questions about the methodology you used is, can you outline how you quantified the collagen content in the muscle?
Yeah, okay, it's so to have a broader answer. So I would say we had a little bit different technical setups for the Duchenne and SMA study and also for healthy volunteers, but more or less we used about 12 to 14 wavelengths in the spectrum from like 660 to 1200 nanometers. And we used a setup which was provided actually by the manufacturer and a spectrum which is also implemented in preclinical systems, and used actually this software to then spectrally unmix it. And what you then do is to really outline a specific region of interest. And what we think and what we did, we excluded the fascia directly, which is around the muscle, which we know which is rich in collagens, and we only measured directly inside the muscle. So this is intramuscular fibrosis, or let's say signals what we got. So this was the approach we had, but we are still testing on different things. We also try to unmix it for other chromophores. But on the collagen side, it was like, to wrap it up, 12 to like, let's say 14 different wavelengths in a little bit altered setup for these two studies.
And relating to the sampling you did for the regions of interest, did you image the entire muscle groups, or was there a reason why you imaged certain sections of each muscle?
Yeah, so what we did there was kind of a standardization. So we described different distances which we measured from anatomical points. So it's like 1/3 from such a dedicated position. And this is how we standardize. So we really measured distances and like took a cross section there. In the data I showed you very later on this standardization study, we kind of try to image different planes in shorter distances, where we kind of try to get an information if there's very heterogeneous information in one muscle. But we decided for these first studies to have like one representative plane, cross-sectional plane in one muscle group, which were these defined groups I showed you. And it's always related to one specific measurement between two dedicated anatomical points.
Okay, so the ultrasound probably is useful for guidance, I guess.
Yes, for sure. So for this, you definitely maybe need a hybrid system. So hybrid means you need to combine an ultrasound or B-mode information, which is called reflectance ultrasound computed tomography in this like technical words in MSOT, but you need to kind of fuse it together with your optical image so that you as physician have a guidance that you assure to some extent that you image the same muscle groups.
Okay, again to muscles, are there any muscles that you had to exclude? And this relates to another question, which is, would there be problems imaging, for example, thighs in the adult or obese boys?
Yes, so this is a very, very good, and I really appreciate this question. So yes, the most impact or the most problems from this imaging technology comes from depth penetration limits, I would say. And if you're developing a system which is maybe also in the future ready for clearance, then you have to see that all membranes and everything is ready to such an application. And this maybe limits also your depth penetration. And I would say there are muscle groups which are much more easy to image. And this is really, really true for the SMA study, because their muscles are very, very small, and fat tissue is much more prominent. And I would there say that upper extremity is maybe easier. So biceps muscle seems to be easier than muscles on the lower body where much more fat is present. And if you have like huge, like subcutaneous fat, then you have problems to detect fat inside the muscle. So I would say upper extremity works much better in SMA. We didn't really recognize this to this extent in the DMD study, but for SMA, I would say there are restrictions on the legs.
Okay, so another slightly technical question is, so for example, in the SMA study, did you segment out the subcutaneous fat layer and just look at lipid in the muscle?
Yes, the answer is yes. So it's just intramuscular lipids, and yes, we have to say there's to some extent this huge variation because you have like different signals for subcutaneous fat, and this kinds of absorbs some of the signal which maybe could come from the muscle. So we are not really confident with the signal, and we have also seen fat signals in Duchenne boys, but also there we need to say it's limited by absorption from subcutaneous fat. So I think there's much more to do if you want to account for the subcutaneous fat and to measure really fat inside muscles. You need to develop different strategies, I would say.
Okay, I'm actually going to jump in here and answer a question which we've had by quite a few people, which is that yes, this system is available for imaging mice. The system used here was the translational stroke clinical system, so is designed for larger subjects, but we do make a scanner which is designed for imaging mice. I'll stop talking now and get back to the proper questions for Ferdinand. I like the sensitivity and specificity, but is there a limitation in terms of scan length?
We described it a little more precise in our appendix in the paper. So if you image all muscle groups, and you have to say we imaged like biceps, flexors, and also 2 muscle groups at the legs on both sides, which makes 8 muscle groups. We had total scanning times of or exposure times to the laser, I have to say, which are around 6 to 7 minutes. Yeah. So it's more a question of a few seconds. So if you want to have a huge spectrum, it's maybe 30 seconds. And if you want to restrict yourself to maybe, let's say 12 wavelengths, and you have a 25 hertz laser, so it's less than a second what you need. Maybe you want to have more frames to have a little bit more stable images, then you have only a few seconds of imaging. Yeah. So I would say, as compared to MRI, it's still very, very short times. You need to expose a muscle to the influence of a laser, and compared to that, you have maybe 20 minutes or a little bit lower maybe for a leg in an MRI. So scanning times, I think it's not really the restriction with this method.
Okay, again, back to the technical detail, is you used wavelengths above 1000 nanometers. Was there a reason for this?
So the expectation we had is that we think, and I showed this at the introductions, was very short, sorry for this, is that we get absorbance from some of these chromophores, and this is very much collagens, also water, which happened in these higher wavelengths. And it's not so much if you image in the near infrared, like one window, which is maybe until 900 nanometers, hemoglobin's very, very prominent, and we actually wanted to avoid this to some extent, and this is why we used these wavelengths in the study to get a little bit more confident view on the collagen side and to get some peaks from collagens which are way beyond maybe 900 nanometers.
Okay, thank you. What center frequency on the transducer?
So if I'm correct, and Tim, you have to correct me if I recall it right, it's 4 megahertz.
4 megahertz on the 2D.
On the
yes.
2D.
Yep.
Yes.
And I'd have to check the methods, but I think either 4
Nine,
or 8 megahertz.
It's 4 megahertz.
Yep. On the 3D cup. An interesting question about safety in young children is, this is obviously a laser platform. Are there any safety issues with using this on younger children? Has anything been reviewed so far in clinical studies?
Yeah, so we had a huge discussion about this, and also iThera provides some information about this. So concerning the laser, you get not over these skin limits, anti-skin limits, so it's beyond these limits. And also we think from this just experiences we have here with children so far, we have not seen any effect on the skin, no reddening, no harm to the skin, which would be the first side of damage, but you're way below the limits, and we don't think there has been any effect yet. So the most advance recorded from us relates to ultrasound gel, which is cold, and also to the safety goggles which you have to wear, which are not that comfortable. And I think these are the most issues, but concerning the laser exposure, we did not see any problem, and we did not record any adverse event there.
Okay, and I'll finish with one final question, which is, lots of people have mentioned this in the comments. Very nice work, Ferdinand. Can the approach be used to monitor treatment progression, assuming a treatment exists for the disorder?
Yes, so this is what I try to show you now, this field, and I'm very grateful for this. We can now offer treatments for our patients. I only showed this example for SMA, so we have genetic intervention, and I hope that you can use it. We only showed that we see progression, and just from this assumption, I would hope that we can do it. I still don't know if we are early enough with our treatment to really change the course of the disease and to really change the prenatal onset maybe of the disease, and to save enough muscle tissue and to see developing a normal tissue. But just from my side, I would say I would really hope to see this, because the differences between diseased and healthy are so big. I would just expect maybe something in between, which then is treated. But from now, we don't have a longitudinal treatment study. I can't say this. So this would be interesting in a preclinical model, which will be very complicated. But also, I think there is now a chance to also bring this to a clinical setting with treatment which really alter the course of the disease.
Thank you, Ferdinand. And I'll also say a big thank you to all the people who attended today and who stayed on, and for making some really interesting questions. It has been mentioned, we will put this webinar up on YouTube in the next day or so that you can review it in your own time. And I'm sure Ferdinand would welcome any contact for people to talk to him specifically about his work and objectives and what he presented here. So again, thank you very much. Thank you to Ferdinand, and I'll hope to see you all in the not too distant future.
Thank you, Tim.